there was a reduction of habitat that limited expansion. So two things were kind of making that the species richness was quite comparable while the composition of the species had changed. For conservation purposes, well then maybe what we need to do is, because it's hard to change climate, but we can maybe restore plants and habitat to favor the maintaining of the species. Okay, so now I'm going to change gear and bring you to an analysis of malaria incidence. This is to show the dynamic system and how can we study that in a space-time context. So I need to make a little parenthesis here. So basically, we are thinking often in connectivity terms, talking about the structure of a landscape, and we talk about how things can move from one pond to another. So that's just the layout of my habitat. And here I have a frog, and maybe how the frog can move in the landscape is a function of its dispersal difficulty. So the functional connectivity for the frog is a function of what is available as habitat and its behavior to movement. But we can extend that to disease spread as well. So here we can say, well, the disease spread is a function of this actual connectivity and the encounter rate between the different organisms. So in this case, of malaria would be human and the mosquitoes. So we can think of this interaction is spreading the disease in a landscape, and we want to study that. So we did this, this is in collaboration with Marie-Josée Fortin and Geraldine Jordan, former student in my lab. And we were looking at how can we explain this dynamic of malaria in Venezuela. And to do so, we developed what we call a kind of state transition index to measure this temporal dynamic. So the study areas, this is Venezuela, was this little region, and the study area was 332 square kilometers. And the processes that we were studying is the spatial heterogeneity in terms of topography, the connectivity between human and the mosquito, and the interaction response of the disease. So here is the map of the region. So let me explain to you what we see. So the kind of little pattern here, those are marsh wetlands. The triangle are the mosquito habitats along those marsh. The black line are the main roads, and the circle are villages where the size of the circle is proportional to the incidence of malaria for the year 2002. The information of malaria was collected at the hospital level, and what we know is a weekly incidence. So we have weekly information. So this is the year 2002, and if we compare to 2007, it was too quick, you could not see any things, but basically there's some changes. Oh, and I forgot to continue my exploration of my landscape. Those are more mountains in the background. So this is flat, and this is more terrain. So let's say that we focus on this town here. In 2007, the incidence was much higher. So we want to study how can we characterize the fact that some areas seem to have low incidence and some have high incidence. So first of thing we tried was to explore your data, always map your data. So we use this statistic called the local Getis statistic that allow us to find hot clusters of malaria and cold clusters of malaria. What do I mean by cold cluster? I mean a region where the incidence of the disease was less than the mean, and the red areas are the location where it was higher than the mean. So in 2002, we see that in the mountain, this incidence of malaria is very smaller, less incidence than in those regions in red here. The circle in gray indicate that this is not significantly different than random. There was incidence, but we cannot tell if it's really exceptional or not. In 2007, we see no cold spot. We see only a hot spot of transmission. So we wanted to explore this dynamic a little bit more. And to do that, we use what we call a kind of match-mismatch type of idea. So let's think of two towns, so two villages that are next to one another. There's two possibilities, is that the both towns don't have the incidence. The two towns are having malaria incidence. One has it, the other one don't, and the other way. So those are four combinations, and we are going to look at how many times through each week over the 7 years of the data that we have, this type of context occurred. But first we need to decide where to put those kind of link between the towns, the villages. Oops, the other way around. So we have developed those Voronoi polygons. So this is now each town, and we have those kind of polygons that segregate them. And for each village, we know who's the neighbors, so we're going to look at those mismatch at those edges. So those edges will be, for those of you who have read my book, and the Delaunay links between them. So we do that for with the study in 2001, we had only 47 weeks. But then we did that for 2002, 2003, 2004, and you can see that through the years, different mismatch occur, but the mountains region have always less differences than along the road. Then in 2005, 6, and 7, so then we regrouped all the years together. We did a randomization procedure, and we were able to test the significance of those mismatch. And in green, those are the regions that are here in the histogram, that would be those guys, where the incidence, the mismatch between village is higher than by chance. And in red is the lower tail. It's occur less likely, less often than by chance. So we can differentiate two locations, and the one in black is not significant. So we can see this dynamic, but I promised, I claimed that I was going to show you a state transition index. This is not a state transition. I just clapped all my time together. I'm lying. So I need to do that. So how am I going to do that? I'm going to do a 3D tic-tac-toe, where I'm going to say, well, there's three possibilities we tested. Either this village is always having an outbreak and the next one does not. It's partial oscillation or spread, that this town has an outbreak and not the next one, and at the next time period week it would spread to the other one. And or there's a flip-flop in the sense that a week was sufficient that this group of people that had malaria are now healthy, but the next village got it. So we compute those three state transition index to incorporate both space and time. So when we do that, we have those three panels for the three different statistics transition. So here we see the same thing, darker, it is more of those permanent condition that is always these boundaries that are occurring more than by chance, and the other ones are maybe just random one. The spreading from one village to the next is occurring mostly everywhere, and this flip-flopping is maybe at the center. So what we can conclude from this study is that those state transition index are useful to identify the behavior in space of the disease spread, and maybe tell the authority that the larger towns need to be controlled more for their spread to smaller towns. So this was a way to capture the spatial temporal variability in the system. Okay, I have time to race to the last example that I'm going to share with you. So this study is about the genetic connectivity of Boreal Chorus Frog in Colorado. And basically, we want to see, is the animal moving in the landscape, and is there some change of genetic material? So this is work in collaboration with my current student Alexander Watts and a group of people. The data comes from Melanie Murphy, and we were looking at this kind of habitat connectivity and the fluctuation of the habitat. So I'm again trying to see how the fluctuation of the habitat will influence this connectivity. So we wanted to see, here the habitat is a pond, and how this is going to affect my genetic signature. So the same way that we had previously the connectivity that is due to the dispersal ability, the disease spread, we can have the genetic connectivity that is a function of the actual connectivity plus some gene flow. So the little frog here, can it move from pond to pond, and the frog that we are going to study is amazingly small, so the dispersal ability is very limited. So the processes that we are going to study is a mix of metapopulation, local weather, connectivity, land use change, and climate change. So the study is in Colorado, and there was this sampling of 18 breeding ponds. So those are the letters that you see. This one is very isolated. 328 individuals were captured. They had the DNA sample to have then 12 microsite loci taken, and some environmental variables were measured: the precipitation regime and the relief, the topography, topology between the ponds. Why did we select that one up there that's very separate from the others? There was a study in 1960 that had previous knowledge of this 4 ponds that were in this area, so 3 here and this one. So we wanted to see the changes between previous time periods. So if I was to look at the distance matrix of connectivity between all the ponds, that will give me this kind of spaghetti. We wanted to try to analyze this relationship between genetic and the environment. To do so, we use gravity model, and we look at the dispersal ability of the species, the influence of each pond, and the land cover between the ponds. So the gravity model, for you who have no clue what it is a kind of a regression that is a bit complex when you look at it. Your response variable in this case for us is the genetic distance. Then we have the actual decay function of dispersal ability of the species between the ponds. We have the attraction power, so bigger the pond, more likely the species will stay at one point or go to another pond. And then we have maybe this in between, the landscape characteristic that will make the frog to have difficulty to move in this area. So we decided that, we'd like to know about the functional connectivity. So we developed 3 types of network of connectivity. So the first one is more the functional one that I mentioned to you. So those are the 18 ponds. And we say, well, we're going to look at the dispersal ability of our frog. And I know it's written very small here, but it's 600 meters. So those little frogs don't move much. They are very tiny. And these are the buffer of 600 meters around each pond. So you see that there's maybe a cluster here, a little cluster here, and these 2 are very by themselves. So this connectivity is really reflecting what the species is able to do in the landscape. So then, I thought I had removed this slide. Well, okay. So again, just to show you. So we wanted to reclassify our ponds as a function of the precipitation regime, because the one in reds were more established pond, more permanent ponds, while the one in yellow were in fact, good ponds as well, but could fluctuate depending on the precipitation regime. And the blue one exists only when there was a wet year, because they dry up. So we have a change of those ponds through a time series. So we wanted to explore that. So what we did is we look at how many years in a row a pond is going to be wet, so there's possibility of maintenance of the population, or only those will be maintained because they can sustain a drought, and so on and so forth. Then we measure the connectivity of the animal. So this is, you see how small it is, by different matrix that were developed by Santiago Sora, and basically the connectivity is the mix of the ability of dispersal, here 600 meters, so we call that the flux, versus how big the ponds were, that's intra, and how many times a connector, so without this pond H, I cannot go from one location to another. So those are kind of stepping stones. So we can divide the connectivity on those three different components. And when we do that with our functional sample network, the panel on the left is the circle are proportional to the amount of connectivity that we found. So this one is by itself but has some connectivity. These are connects but have very low connectivity. And then we look at what proportion of the connectivity is due to the flux. So that's the green portion of the pie. So most of them, they are the connectivity is due to the possibility of movement, the flux. And this one that is by itself is mostly intra. It's a big pond, enough big that the species can live there. And this one is kind of in between. So that was our network that is just due on the functional ability of the species. Then we say, well, through the summer, the ponds can shrink. So there's some in the precipitation regime. So we will have some years where we have more ponds, more connectivity, and some years where we have less pond and less connectivity. So we developed what we call a temporal connectivity index, and we use for that some snow-covered data set for 33 years, and this is kind of an index of how much water there is in the area. So we have some ponds that are going to exist only in dry conditions where there's less than 25 inches. Some that will be present only when there's wet years, more than 25 inches. And then some that don't change. They are so big that the amount of precipitation is not so important. So this is already what I showed you. So those are the yellow one will be present in dry years, the blue one only in the wet year, and the red are always there. So we had each year a flip-flop. Sometimes it was dry, sometimes it was not dry. So we have a composite of all those years that we took, and we look at, so that's the dry years and the wet years. We look at the connectivity, and now the picture changed completely. So the connectivity is the highest in this sector, and this one's still there, and the other ones have not much connectivity. And most of the connectivity is due to the size of the pond. That's what maintains the species there, is that the pond is big enough and it can survive there. There's a few that has some flux connectivity, but this one is by itself and big enough. Then we say, well, we're a little bit lying here. We have only 18 ponds. I mean, there's other ponds or streams in the region. There's in fact 110 of them. So we decided to have those kind of stepping stone intermediate wetlands that maybe the frogs are using, because we don't have the genetic, but they are using them. So we build this kind of new network. And here we see, again, that some are, the connectivity is due to the size of the ponds, but there's more connector than we didn't have any connector before. So they are using those intermediate ponds. So we put all that in our gravity model, we shake and bake, and we had those results. So the best model was the last one I showed you, where they are using the stepping stone, so we call it suitable habitats. the amount of topography, relief between the site was important, and the amount of precipitation. So that was the best model that explained the genetic components, genetic variability that we have. Just for showing you how things they improve if we use the temporal. Here I put age to say just the years where that was very wet. So we just use years of imposing the connectivity due to ponds that are in the wet years. So the difference AIC was 5. If we were looking at our functional connectivity, the sample one, it was close to 6. And the temporal one that we worked so much for, that we had the fluctuation of low years and high years, was not very good, was 8. So at the end of the day, I think the species can make it because they are using those stepping stone ponds in the region. So this is a case where we were able to show that the change in precipitation will definitely have an impact on the ponds, and that the connectivity is due to the stepping stone. So I hope I showed you that there's a series of ways in which we can study ecosystem, landscape, that the framework of spatial ecology allows us to look at processes, the patterns that emerge from that, and we can model them. And I would like to acknowledge all those bright students that are in my lab. So Ilona, that just graduated, and this work was in collaboration with Kathy Lindsay, Alexander Watts, Melanie Murphy, Aaron All, Dan Burt, and Miller. Oops, I made a typo, don't tell her that. And Geraldine, she's not on the picture, she's a former student of mine, and Maria Virginia. Thank you.